So we can go back and we can look to see when two species diverged by looking at genetic differences between those species. We can use those to reconstruct their evolutionary histories, or what we'll call phylogenies. This is basically like a little family tree or a branching diagram. All of the branches are going to represent your lineages over time, and you may have seen diagrams like this before when you're looking at, say, the tree of life. Groups that consist of an ancestral species and all of its descendants are called monophyletic groups. We can look at reconstruction of the amino acids of cytochrome C in a phylogenetic tree. Remember, cytochrome C is a component of the electron transport chain of mitochondria. And what we can see is that it has evolved very slowly in a whole variety of organisms. The more closely related two organisms are, the more closely related their amino acid sequence will be. And the more distantly related the organisms, the more amino acid sequence differences there will be. So here's a chart of humans and a whole bunch of different species. And what we can do is compare the amino acid differences between humans and these different species to try to reconstruct a tree. So, okay, of course, humans is 0 because that's what we're using as our baseline. In this particular case, chimpanzees actually have 0 amino acid differences from that. Rhesus monkey, also very similar at 1. Rabbit would be 9. Even all the way up to a moth with 24 differences, or a yeast with 38 differences. It seems like a lot, but it's really not when you look at the overall structure of these organisms. So when we calculate this using this minimal mutational distance, minimal mutational distance is just the amino acid differences in a protein, we can use this information then to construct this phylogenetic tree showing the relationship of these various different species. And it gives us sort of a distance of how long ago do we think that these species branched off from one another. So we can look, human and monkey are very close, horse and donkey very close, chicken, penguin very close, duck pretty close to that, pigeon pretty close to that, turtle pretty closely related as well, snake, tuna, geese and bread mold pretty distantly related from a lot of these others. Molecular clocks are going to measure the rate of evolutionary change in terms of amino acid or nucleotide sequences, and what we're going to assume is that these changes or mutations are going to accumulate at a constant rate over time. We're going to assume these organisms were not exposed to any weird chemicals or UV radiation or anything like that, but that this overall mutation rate is going to remain constant over time. And what we can see on the next slide is a phylogenetic molecular clock looking at influenza A hemagglutinin gene. So this is going to be one of the proteins that's going to be on the coat of influenza virus, and it's going to be responsible for getting that influenza inside of the cells. And what we can see here is as the number of nucleotide substitutions goes up, we can figure out the year that they were isolated, and it goes up pretty straight line. You can follow these individual isolates here and how they might be related to one another. Neanderthals, a lot of people are interested in them. They lived in Europe and Western Asia about 300,000 years ago, and they disappeared about 30,000 years ago. So in the case of evolutionary time, that's really just a blink of the eye. We do know that they coexisted with modern humans. We call them Homo sapiens versus Homo neanderthalis in about 30,000 years in several different regions. And so people were curious, are we descended from Neanderthals? Are they a completely separate branch? How is this working? And what they did was use this mitochondrial DNA from the Neanderthal bones, as well as more than 2,000 present-day humans. And their conclusions were that Neanderthals and humans have a common ancestor. The Neanderthals were a separate hominid line and did not contribute mitochondrial genes to Homo sapiens, although there may have been some interbreeding going on. And so you can see here how closely related Africans and non-Africans are. You can see compared to Neanderthals, it's all of humans are going to be distinctly related from Neanderthals. And you can see here as a similar sample, you've got 5846 modern humans, and they're all going to be in this tree here, and they're a completely separate branch from these Neanderthal samples. These happen to be two samples that were found in two separate caves. You can see chimpanzees, also related, pretty closely related, but we do have just a distant ancestor in common. We didn't evolve from chimpanzees, but we had a common ancestor with them. The Neanderthal Genome Project resulted in a draft sequence of this Neanderthal genome. What we found is that it contains 3.2 billion base pairs, just the same as ours. It's actually 99.7% similar to our genome. Both the Neanderthal genome and our genome are about 98.8% similar to that of the chimpanzee.